It's okay. So the title of the talk is Confocal Microscopy, a new dimension in scientific imaging. But I personally feel that confocal microscopy is like a rainbow revolution in the microscopy. Now we are going to talk about the imaging. So before we start imaging, let's understand what is really important in imaging. Can you all see my complete slide? Because I'm not able to see the slide in totality.
Ma'am, we can see the slide with
Okay.
the title and the definition.
Now it's I see into. Um, Okay, so when we talk of imaging with microscope, what actually we mean to do is microscope is an instrument which is designed to make finer details visible. And to achieve this, there are three important principles that one need to look into. Let's just look at it one by one. What is important is you need to magnify the object. So the small object should be magnified to such an extent that we can see it with our eyes. Another important point is resolution. That means small detail of the objects, as you can see resolved over here, should be distinguishable when we magnify the image. And third most important is the contrast. Like the image which is magnified and resolved must be visible against the background. These are the two examples I have shown you. Now, keeping this in mind, we can go ahead with the imaging. But before that, I would like to just give you a reminder to the parable. I'm sure most of you must have heard about this elephant and seven blind men. These seven blind men, when they were asked to describe the elephant, depending on whatever they have touched, they described it as a rope, as a pillar, or as a wall. And this is because I think their perspective was limited since they couldn't see the elephant. So, what we need to do is have a complete perspective of any object that we are going to see. Then only we can actually say that seeing is believing. If we don't see everything in three-dimensional, as a three-dimensional object, it will not be possible to get complete information about any object. And this 3D imaging is possible only with a laser confocal microscope. So I'll be talking on laser confocal microscope, then multiphoton excitation microscope, which is a kind of advanced version of laser confocal microscope. And then we'll discuss few of the applications in the cell imaging that are possible with laser confocal microscope or with the multiphoton microscope. But before we actually turn into or touch the topic of laser confocal microscope, I would say let's brush up about the fluorescence, because most of the imaging that is done with laser confocal microscope is in the fluorescence mode. So in this mode, we need to know what is fluorescence so that we can really operate it to the full extent. So as you all know, fluorochrome is the substance that absorbs light at a particular wavelength, get excited, and subsequently it emits light of a longer wavelength. So for example, you have a dye like FITC, I'm sure you might be familiar with this. So it absorbs a blue light, gets excited, and then it emits energy, which is a green light. It's of a lower energy, and that's how after emitting green light, it comes back to the ground state. Always remember that longer the wavelength, lower the energy, shorter the wavelength, higher the energy. These are very important principles that one should remember when we are talking of the fluorescence microscope. Now, how does fluorescence microscope actually work? So this is a kind of optical path for the fluorescence microscope that I have shown. What you have as a illumination in fluorescence microscope is a mercury arc or xenon arc. They have multiple wavelengths that are emitted. So what is required is you need to choose the wavelength depending on the fluorochrome that you are using. Say for example, now we are going to deal with FITC. So FITC needs a light which is blue, then only it gets excited. So what is done is you put the excitation filter which allows only a particular light to pass. So here it's a band pass 490 plus minus 15. That means it allows wavelength 490 plus minus 15. That includes blue light. So blue light is passed through this filter while rest of others are totally blocked. When this light hits on a dichroic mirror, which is at 45 degree. Now this is dichroic mirror 510. That means it reflects the light below 510 and allows the light above 510 to transmit through. So naturally, when blue light hits this, it reflects it down through the objective on the object where the specimen is sitting. Now the light or the fluorochrome, that is in this case FITC, will get excited with this blue light and it will emit green light. Now green light is in the range of 530 to 540. So naturally it will be transmitted through this dichroic mirror and it will hit the emission filter. Now emission filter selects the light that we are interested in and then sends it to the detector. So these are the bandpass filters. Suppose you have more than one fluorescence, then you can actually split that, or you take the one which you are selected in. Say in this case, the green is passed through. So if you look at the microscope, what you actually see is you have a light source, then diaphragms through which the light is directed to the excitation filter. This is the kind of block that generally we see in the fluorescence microscope. Then you have a dichroic mirror and an emission mirror. Like in this case, the green light is exciting the object, and then the red light which is emitted can be seen through the emission filter. So once this is clear, why do we use this? Because when we have a fluorescence microscope, we do get a very colorful picture. I'm sure you would like to see such kind of colorful pictures, but remember now in this picture, you see nucleus, you do see fibers and some granular material, but these fibers are not seen clearly. There is a haze in the picture. Now, this kind of image is not really giving all the details that are present in the cell. So we would like to have a very clean image. We need to have a better resolution of the image, and if possible, even magnify the image. So to do this, the concept of laser confocal microscopy was first put forth by Marvin Minsky in 1958. And in 1957, this concept was patented, while in 1970, the concept of laser confocal microscope was established by Yale. And later on in 77, Breckenhoff put the term confocal into this. However, it took almost 36 years from 1951 to 1987 for White, Amos, and Fordham to build the actual confocal microscope, which was supported with biorad. So this is how the first confocal microscope was looking at it. I want to show you this picture simply because now the microscopes are much compact. But if you want to understand the functioning, this is the better picture to look at the microscope. And look at the image, the same object is under laser confocal microscope. You would appreciate the clarity, different fibers that you can see, different granules are distinctly seen. And you also get a 3D effect in this. Like this is on the upper plane, while these are lowered in the lower plane close to the substratum. So there is a resolution in the picture. Now, how this resolution is increased? So let's understand what is the principle of the confocal microscopy. So in confocal microscopy, as the name suggests, laser is the source of illumination. So laser light is passed through a confocal pinhole to the object. However, in the path, there is a beam splitter. Now, what this beam splitter is nothing like but a dichroic mirror that the way we saw in the fluorescence microscope. So it allowed certain light to pass through and certain light is reflected. So light of interest that is passing through beam splitter is collected by the object and focused on the collected by objective and focused on the object. Now, black line that you see here is the focal plane of the objective, while the orange line shows planes which are above and below focal length. Now the light which is emitted by the object, it will be emitted from all the planes, but the black line that shows you the light emitted from the object which is in focal plane. So this light as well as those as well as the light which is coming from the planes which are not in focal, totality is reflected by beam splitter to the detector. But on the way, there is a confocal pinhole. So what this confocal pinholes does is, it only allows the light which is in focal plane to pass through, and rest of the light which is coming from the planes which are not in focus is blocked by the confocal pinhole. So what is important in confocal microscope is the light source, which is a laser, which is a point source, and this confocal pinhole, which allows only light which is in focus to pass to the detector. This is just little better elaboration of how the confocal principle works. You can see a blue gas laser which is illuminating the specimen. This is just enlarged view of that. And when the specimen is illuminated like this, what you see is a focal point in the objective, and then light which is emitted from the focal point, only that light enters through the pinhole to the photomultiplier tube. Thus, what we can say is the focal point of illumination is the same as the focal point of detection, that is the confocal. Now let's see what are the different parts of the confocal microscope. You need a source of illumination laser. So what you see is a laser here, which through optical fiber takes light to the scanner head. This scanner head is heart of the microscope, which has all the optical elements sitting into this. This is connected to the microscope. This is like any fluorescence microscope. And then whatever light is coming from scanner head is collected by the objective, which is now acting as a condenser, and it is focused on the object. Now, the light which is emitted by the object is again collected by this objective, taken to the scanner head, and then given to the controller. Now this controller operates scanner head as well as it converts light into the digital image that we can see on the monitor. Now let's go one by one to all the parts of the confocal microscope. Now illumination lasers could be anything. However, most of the time it's argon ion or violet diode which are used as illumination lasers simply because they give exciting line in the blue and ultraviolet region, and most of the dyes are illuminated with blue and ultraviolet light. However, other lights, other lasers are also used, which could be krypton, argon, helium, cadmium, so on and so forth. Then this scan head. This scan head has major components like reflecting mirrors, excitation filters, emission filters, and photomultiplier tubes, which are for detecting the photons. So let's see how it works. This is a little smaller picture, but I'll try to explain it. Suppose the light is coming through the optical fiber, then there are collimated scanners and then lenses which direct this light to the dichroic mirror. Now this is in the form of wheel simply because there are different kind of filters which can be accommodated in the wheel. This is at 45 degree centigrade, 45 degree, the way it was in the fluorescence microscope. This is reflecting the light, which is a blue light coming from the optical fiber. Then there is a lens to collect this, and this scanning mirror which let it fall or takes it to the objective. Now object is taking it on the object. Now the light which is emitted by the object is a fluorescence which is shown by green line. It follows the same path and hits this dichroic mirror again. So this being little longer wavelength, it just gets transmitted. And again, there is another dichroic mirror. Now, what this dichroic mirror does it, if there are multiple colors, then it divides it into different multiple wavelengths. And at the end of every lane, there is a detector in the form of PMT or arrays. Now, one should notice this small ring in front of all the detector. This is nothing but the pinhole that is required to collect only wavelength which is coming from the plane in the light, from the focal plane of the objective. Now, how does PMT works? How does it detect? Now, the major part or the job of the PMT is to amplify the signal. These are cross sections of PMTs like horizontal and vertical. So what happens is when photons hit the cathode, there is a chain of dynode. So they go on traveling, or rather they're sort of tossed along the dynode chain, and that's how they're multiplied at every hit, and ultimately they reach the anode. Now these signals are then converted or digitized by the controller. So what you see at the end on the computer is a digitized image like this. Suppose you have two channels, like two different colors you are looking at, red and green. Along with that, even the transmission image can be collected, and all these images could be superimposed to form a 3D image having three parameters: red fluorescence, green fluorescence, and the transmitted image. I talked about 3D image, so probably you couldn't see how the 3D is collected. Sort of 3D image is created. Now in this microscope, it's a Z motor. Now what is this Z motor? This is a motor which is attached to the focusing screws, and it moves the focusing screws by the distance that we set in, say 0.5, 1 micron, 1.25 micron, depending on the objects. So it moves this stage in a Z axis. That is why it's called as a Z motor. So what it does is Z motor moves the object which is sitting over here in the Z axis so that the focal point of the laser keeps on changing. We can start from top to bottom or bottom to top. It's up to us. And there is no actual sectioning of the object. But since we are moving the focal point of the laser, only the image which is in the focal plane will be collected. Like this, when we go on moving in the Z axis, we have multiple images, and even the deep-seated signals can be seen very clearly. Now, all these sections, which are optical sections, could be stacked to make a 3D image like this. So we get more information from the same image. At the same time, we can see the deep-seated signals, which could be a weak signal, and we have much better image and more information. The same thing is shown here in a cartoon. What you see is a laser. This is suppose your focal object and a focal plane. Then you can do line scan with the x-axis over the time, or it could be XY scan that can be done. And this XY scan is nothing but you will get diskettes, and which would be along with the time. So what you get in this is XY and T, because you are doing it several times, and all this could be combined to form a 3D image to give a more information than anything else. But does it give you little more information? Let's look at it. But before that, I would like to give an example of a simple fibroblast. Now, these fibroblasts are stained for microtubules, and this sectioning is done from top to the bottom at a distance of 0.5 micron. So in top, you see very little area. That means cell is not a flat cell, but maybe it has a kind of dome on the top. And as we go down, we see more and more area of these cells. As you go almost at the center, you see microtubule organizing centers and lots of microtubules radiating from that. And as we go down, more and more microtubules are seen. Again, when we go at the last point, microtubule number is decreased, and all these sections are piled on top of each other in a same series to get a 3D image like this. Does it give more information? Now look at this. This is convoluria. This is a 2D image of convoluria. You can see there are different objects sitting in this vacuole kind of thing. So what is it? Are they at the same plane? We have no information. But if you take a 3D image, you can get little better information. And it's not just to view, but you can actually measure the distances, because when you have a 3D image like this, you can actually rotate it. So now if you see, this is at a higher plane or on the top, while this is somewhere low in the section. There are lots of things in the center which could not be seen earlier in the 2D image. Everything was looking at like a blank, but there are some even pores in this. All this information is possible because we have a 3D image, and you can also make measurement like X, Y, and Z axis to see what is the distance between it, what is the importance. So that's how the 3D image gives you more information than any other image. So the salient features of LCM, I could say, is confocality and optical sectioning, which takes you to a better resolution, as I showed you in these two images. I hope you agree with that. Now let's just sort of summarize what we have seen till now. So what is confocal? Illumination of a single point in the specimen is combined with imaging of this single illuminated point by a point detector. That's a confocal. Now this confocal system could be two types, digital. So in digital system, actually, depending on the image that we get at different sections or different levels, one can extrapolate, and with the help of computer programming, and for the image, that is called digital confocal. Or it could be a scanning confocal system, wherein actually sectioning is done with the laser, optical sectioning, and then you get a 3D image. Now this scanning could be of the three types: object scanning, beam scanning, and objective scanning. Now when you do object scanning, that means you are moving the object. So there is no variation in the light when you are moving the object, but object being little bulky, it could be little slower than the beam scanning that is done. In case of beam scanning, it is very fast, but then the brightness could vary because this laser is sort of moving very fast. So that's the only disadvantage with beam scanning. And what I showed you was objective scanning, because we are actually, sorry, what I showed you was object scanning, but objective scanning is more or less similar to that, because in this case, the objective is move up and down instead of the object the way we had seen with Z motor. Then source of illumination is a point source like laser. Then objective generally used is with a high working distance, high numerical aperture, and immersion type, so that we can really handle samples like tissue or even some small animals, which I'll show you a little later. Then pinhole aperture is actually the heart of the system, and axial resolution is proportional to the signal intensity. Detectors could be of two types: photodiodes. Generally, photodiodes are used when you need high level signals, like in case of transmission or reflection mode. In case of fluorescence, most of the time, photomultiplier tubes, that is PMTs, are used. So what are the advantages of LCM? You have a better resolution. This is because of the pinhole and the incident light, that is shorter lambda that you use. The blue laser has a very low wavelength, so naturally it's going to add to the resolution. Then lateral resolution that you can achieve with confocal microscope is 0.2 micron. That means it is about 1.5 to 2-fold higher than the conventional optical microscope. The axial resolution is generally dependent on the way we move the Z-axis. Most of the time, people prefer to have about 0.5 micron, so that it gives better resolution in totality. Then another advantage of LCM is that out of focus, that is blur, is totally reduced. Therefore, the resolution is much better. Then optical sectioning eliminates artifacts that arise from the physical sectioning of the tissue specimen. There is no loss of section in this case, which can happen in case of physical sectioning. Then this is a less time consuming because we don't really do optical sectioning, but we are doing it. We don't do physical sectioning, but we do optical sectioning. So there is no separate sample preparation that is required for this. When since we are doing optical sectioning, we can go deep inside and even the weak signals could be detected. And visualizing a three-dimensional image is the most important advantage. And this can be used, as I said, in two modes, fluorescence mode or reflectance mode. However, like every coin has two sides, there are some limitation with LCM. One major is the phototoxicity. Since we are using laser, which is with high power, it might lead to toxicity. It would also lead to photo bleaching because we are exposing the whole sample to the laser. Another important thing is there are rather limited number of excitation wavelengths with lasers, and lasers are much costlier than any other source of illumination which is used for the fluorescence microscope. So there was a solution to the problem of phototoxicity or photo bleaching, and that was nothing but a two or multiphoton microscopy. This concept was first put forth by Maria Gopart Mayer in 1931. So what is this concept? I'll just read out and then try to explain. Given a sufficiently high density, two or more photons could cooperatively excite an electronic transition that would be normally require a single photon with twice as much or more energy. That means two photons, when they actually of a lower wavelength, say for example, X energy, and they hit, what happens is when they collide, there is a kind of burst and the energy is added up. So you get a high energy wavelength. So for example, if you have red laser, which has a longer wavelength, but less energy. Remember what I had told in the beginning. So longer the wavelength, but less energy. But when the laser collides because of the high density, what you get is the energy is say double. The X energy becomes 2x if there are two photons which are colliding, and naturally the wavelength is much lower now. Now, this two photon excitation was discovered by Kaiser and Garrett in 1961. This was just a conceptual thinking. While in 1964, Bradley and Singh, they came up with a three photon excitation. And ultimately, the microscope with two photon excitation for optical sectioning was thought of by Denk, Strickler, and Webb in 1990. Let's have a quick look at what is the difference between LCM and its multiphoton. So as I explained, in case of laser confocal microscope, you have a blue excitation which excites the fluorochrome. It goes high and then it emits light of the higher wavelength. At this illumination, the whole object is illuminated. So naturally, there is a photo bleaching of the total area that is exposed to the laser. In case of two photon, say for example, red laser. So these are pulse lasers. So they come as a wave. So when they collide, what you get is a higher energy, that is blue light. This blue light excites fluorochrome and green energy is emitted. Now, the collision point cannot be like a whole block. So it's a particular point. It's a very well controlled reaction. So what happens, say for example, this is a point of collision. So only in this point, there will be a blue light. The rest of the sample is saved by exposing to the excitation wavelength. So naturally, whatever is illuminated will only show photo bleaching. The rest of the sample is saved. So there is no photo bleaching. Rather, photo bleaching is very much limited to the area of excitation, which is a point source. The same thing is explained here. I hope this will clarify it more. You are familiar with this picture of the confocal microscope. Now, what happens in case of multiphoton, we are using solid state laser, which are pulse laser. And these lasers, when they excite, what you see is actually not the whole field illuminated, but a small point here. So can you see this small point? The picture enlarged is seen here. And what you have is the rest of the sample is protected from exposure to the light and therefore from photobleaching and phototoxicity. If you try to image this, both the illumination, this is how it looks like. Now this is a fluorescent substance. What you see here, a dot-like thing, is the one which is excitation of the multiphoton. While you see this line, that is because of the laser confocal microscope, the whole sample length is illuminated. So if you try to compare advantages and disadvantages of this, the advantages of multiphoton or two photon microscopy are like this, like they have small excitation volume, so less photobleaching, longer wavelength. We are using red wavelength, so less phototoxic. Then light scattering is too much better, in the sense 2 to 3 times better depth of imaging can be achieved because they are of longer wavelength. Then there is a better separation of excited and emitted light, because you're not actually using the excited light. So there is no question of actually separating them. So there is no need of pinhole in this 2 photon microscope, and there could be less absorption of incident light being longer wavelength. However, there are some disadvantages too. For example, there are some pigments like melanin or plant pigment which absorb this infrared, and therefore they might burn in the beam. There could be absorption of water because of this, and the operation is a bit complex because you need a pulse laser, and resolution could be less because we are using longer wavelength. You all know resolution is directly proportional to the wavelength. So naturally, if the resolution you're using longer wavelength, the resolution will be less. And then cost of pulse laser is another major issue. They are much costly as compared to normal lasers. So where this technology can be applied. So I'll give you a few examples by giving images and just run through various applications that we can have. Say if we are talking of the cell biology or zoology, any life sciences, then we can look at different cell structures. We can also look at various compound. We can look at drug uptake, physiological responses of the cell, like cell motility, cell cycle, then molecular interaction, diffusion of molecules, co-localization, cell signaling. So what I'll do is I'll just give you few examples of this. Uh, I'm not running through because anyway this will be covered. But when you want to prepare a sample, what kind of sample is required? As I said in the beginning, you don't need a special sample preparation for this. And both live cell as well as fixed cells, both can be used for confocal microscopy. When it's fixed cells, there is no question of doing any additional thing. The way we process it for fluorescence microscopy or other microscopy, we can process it. But for live cell, for the probe to go in or the dye to go in, what you need to have is a ester group on dye so that it can enter through the cell membrane. Alternatively, one can microinject the probe, or the probe can be entered as an endocytosis or infusion as a lipid-soluble moiety. Now, almost all the cell structures as indicated here can be seen through the seen with the help of laser confocal microscopy very clearly. And these few examples of dyes, and all these are commercially available and they have various colors. So we really have a panel of probes, and at a time also one can see all these cell structures in a single cell when we have a good combination of the probes that we use from the list given here. Just giving you examples of cell structure here. Now what you see is a blue nucleus and red nucleoli. The Golgi apparatus, which is seen in green around the nucleus, and what you see red is actin stress fiber. While in this case, stress fibers are green, microtubules are red in color, then nucleus in blue, and wherever there are focal adhesion points, you see this yellow color because there is a overlap of microtubules as well as the actin element. Then mitochondria in this can be seen as a red. While in this case, what you see is an endoplasmic reticulum, which is spreaded throughout the cell in the green color. Then peroxisome can also be seen. So n number of cell structures can be seen in combination or alone. If you want to give a treatment of the or during drug discovery, so what is the effect of the drug that you have on the target? That can also be seen. So one example given here is drug targeted towards actin, because actin is one of the dynamic molecules. So what you see here is this is NIH33 upper panel, and lower panel is MCF7, which is epithelial cell line of breast cancer origin. So untreated cells show very good actin filaments here. However, when you treat them where the target is actin, all the actin filaments start dissolving, and you don't see the network of actin that can be seen, which confirms that your drug is targeting on the desired target actin. Then cell signaling can also be seen. It could be like just signal coming from growth factor, hormone, or some G protein coupled receptor, enzyme link receptor. I'll give you one example. Now, in this case, what is done is there is a fluorescent formyl peptide, and we have studied its binding to the formyl peptide receptors on this cells called green 9815. Now, this is done in fixed cells. You can also do it in live cell to see how the receptor ligand interaction occurs. So, in the beginning, now one thing like for this as well as subsequent slide, most of the times it's dark blue, light blue, followed by further light blue are on the lower scale, while yellow, orange, and red on the higher scale of the fluorescence. So wherever you see red, that means it's intense fluorescence, and light blue means very low level of fluorescence. So in first 30 seconds, very little of the ligand can bind to the receptors which are present on the cell. So what you see is just the decoration outline of the cells. As the time progresses, you see more fluorescence on the edges, and subsequently you see it in the cytoplasm. That means receptor had bound to the ligand, and the ligand is taken in along with the receptors, or just receptors could be recycled back. We don't know yet because we are at the 10 minutes time point. But as the time progresses, you can see that receptor is polarized here. And later on, almost the whole cell is full of receptor. This shows like similar, we can do it in the live cells also and follow the receptor ligand interactions, which are very crucial when we are using certain molecules for treatment of the cells. Then various cell physiological responses, like it could be ions like calcium, magnesium, or pH of the cell, membrane potential, neurotransmitter release, that also can be studied in live cell imaging. This is one example of calcium. This is a oocyte. Again, see, first two, these are all 3D images. In the first two cell images, what you see is calcium, which is at the basal level. It's at blue and green color. When it gets excited at one end with a Q, what happens is you see a tinge of red indicating suddenly high burst of calcium in this region. And then this calcium wave keeps on spreading, giving red, orange, yellow, goes on, green. And over the time, the whole cell is full of high concentration of calcium. Like this, you can monitor lots of ions by using different probes. There could be oxidative reactions which are very important, like superoxide, nitric oxide, or glutathione, hydrogen peroxide. We have almost for every enzyme that we generally want to study, have different substrates which can be giving fluorescent results, and we can look at them either as a direct intensity of fluorescence, or some reagents are such that you can even ratio them down so that you are more sure about the fluorescence intensity than having a single color. Intracellular trafficking of molecules also can be seen in live cells. These are live cell examples. These are cells with actin EGFP, and these are with plain EGFP. You can see there is no structure in this cell, while here you can find filaments of actin. Now, this is a secretory peptide which was tagged with GFP in HEK cell. Initially, the peptide was seen all over the cell, but the minute we stimulate cells, the peptides start going towards the nucleus and gets concentrated around the nucleus. Now, in this panel, it's histone 2B, which is labeled with GFP. As you can see, it is bound to the nucleus and all the time remains associated with the chromosomes, even during the division. In addition to these dynamic events, even the cell behavior could be seen. And most of the time, for this, we have to have live cell imaging. So it could be endocytosis, exocytosis, cell secretion, cell death, cell motility. A number of such reactions can be seen. The first I'll explain you is the fixed cell reaction, where you see endocytosis in a Drosera, a green dye which is taken up by the Drosera, and as the time progresses, the whole Drosera is filled in with the green dye. Well, in this case, it's a drug delivery. What you have is this LBL intraparticles which are sitting within the Vero cells. This is uptake of oligonucleotides in K562 cells. At 30 minutes, the oligonucleotides are just entering the way we have seen in case of receptors. So you see very weak signal. But as the time progresses at the end of 24 hours, what you see is a concentrated oligonucleotides within the cells sitting almost next to the nucleus. Now let's talk of live cell imaging. Endocytosis, if you want to study. Now what I'm going to play is a slide where yeast is labeled with orange color, and then macrophages are expressing GFP. You can actually see the way this yeast is engulfed by these macrophages. It is taken into a vacuole, and ultimately, naturally, it is going to be disintegrated. So all this could be recorded because you can also have an incubator where you can keep cells in the incubator at 37 degrees centigrade, 5% CO2 on the stage of the laser confocal microscope. So you can monitor it for a longer time and go ahead with observations. Now again, cell proliferation and cell death. This is again example with the fixed cells, wherein the chromosomes are seen as a blue color, then microtubules in green color, and then aurora B enzyme, which is again expressed much higher at the time of cell division. So see, the cells are seen at the different phases like interphase, prophase, metaphase, anaphase, so on and so forth. Here what we see is a nucleus, non-apoptotic nucleus and apoptotic cells nucleus. This isn't fixed cell, but same thing can also be seen in the live cell. Here now cell nucleus is expressing GFP and cytoplasm is expressing red color. That is RFP. Now you can see that in dividing cell, the chromosomes are getting separated very nicely. They are arranged in the anaphase and then separating. So this kind of studies are possible in live cells too. Besides this, suppose if you want to look at the cell motility at different organs, when we all know that when cell moves, whether it's a macrophage or epithelial cell, what we see is the kind of extension of the membrane, which is known as a lamellipodia. There was always a question for all the biologists, what happens? Like we call microtubules or actin as a cellular motors. So do they push cell membrane, or is it that the membrane goes ahead and these microtubules then follow the membrane? So let's see what happens. I'll just play this video. Now in this, you can clearly see the membrane unfolds and moves forward, and these microtubules are then pushing into the membrane. So this kind of studies were possible in live cells and with much better clarity than the conventional microscope. Now in this case, what I'm going to show you is a chemotaxis, which is very important phenomena. So there is a yeah. Now here is a source of chemotactic peptide, and these neutrophils are going towards it, and they're labeled with GFP. So when they're moving towards the chemotactic source in lamellipodia, there is a concentration of effectin, which was expected. And the minute you move source, the cells change their direction and move towards it. This is very important when it comes to the drug discovery kind of studies. And this is a coronin, which is GFP labeled again. And at the time of cell division, whether it's into 2 cells or 4 cells, it is always at the opposite poles that you see coronin. Sometimes you have very weak signals, like if you want to do karyotyping. When there are weak signals, you can use spectroscopy, but uh, if the signals are weak, then again identification of the chromosomes is very different. But then if we have a laser-confocal microscope, you can do karyotyping by enhancing the weak signals with the help of laser-confocal microscope. I had mentioned you about small animals. Now, zebrafish, which is a very transparent small animal, we can study the whole animal on the microscope. Now, what I'm going to show you is a zebrafish heartbeats. Now, what you can see here is, I'll play the movie, but I hope you can see the heart here, the upper and lower chamber. I'll try to play the. Can you see the blood flowing from here and then going from upper chamber, pumped out to the lower chamber here? Another experiment that I would like to share with you is the excretion procedure. We had put zebrafish in the red dye. Now that was just a dye, so it was of no use to the zebrafish. So we were just wondering what will happen, whether it will be excreted or it will damage the zebrafish. And what we found is it is excreted through the digestive tract. Can you see this dye moving? It was first collected. So the rest of the fish you don't see, since only digestive tract is eliminated. But over the time, this whole dye is getting excreted. You can see the dye coming out also in this region. So as the time goes ahead, you can see that constriction is going, the concentration is decreased here. So these are actually happening physiological event in the small animals that we can see in the whole animal with the help of laser confocal microscopy. It wasn't possible earlier. You might be familiar with having xenografts in animal. Now, similar studies we can do in zebrafish. For example, in this case, what you see is a zebrafish larvae, and it is injected with two types of pancreatic cells. The green one, which is non-invasive variety, and the red one, which is invasive type of pancreatic cancer. So this is a day 0 when we had injected these tumor cells, and this is after 3 days. What we see here is this is a published paper by Marx et al. After 3 days, the green fluorescence remains wherever it was. However, the red, which were invasive type of cancer, you see them spreaded everywhere. So such studies are very clearly done with the confocal microscope. You can implant, say for example, another type of cells, Miyapaka, which is again pancreatic cells in the zebrafish embryo. Now you can see this on day 1 where you have injected cells, and after 3 days, you can see that the cells have migrated, and not only migrated, they have increased in number also. So this is one of the advantage of having laser confocal microscope. When you have a thick specimen like zebrafish, you can see it very clearly. Now let me talk about some special techniques in live cell imaging which are useful. This would include localization, co-localization, FRET, FCS, FRAP, so on and so forth. If sometimes it's very important to understand where the molecule is sitting within the cell, especially if it's a molecule which is involved in signal transduction, cell adhesion. So this is an example where we have looked at beta integrin in B610 melanoma cell line. So when cells are not treated, all the beta 1 integrin molecules are around the cell periphery. But the minute you give treatment with some drug, the beta 1 integrin starts moving into the cytoplasm very clearly seen. Similarly, this is ERK1, which is spreaded throughout the cytoplasm in untreated HepG2 cell, another cancer cell line. And the minute you give treatment to these cells, it starts moving towards the nucleus. These studies are very important to understand whether the target is really being targeted or not. What is the effect? What is the signal transduction pathway that is attacked by the target? Another important thing is co-localization. Now, if I show you this image and tell you that okay, the red ones are microtubules, the green ones are actin filaments. So what is the yellow one? So naturally, simple thing, green and red together will give us yellow. So you would say that at yellow, there will be localization of both the proteins together. But is that really true? Suppose they are in the same z-axis, one on top of the other. When we are looking at it from the top, we'll always see yellow color. So it is important to know whether it's a real co-localization or not. So there is another technique known as a FRET. FRET stands for Foster Resonance Energy Transfer. So it is a distance dependent phenomena. There is a transfer of excitation from an initially excited donor molecule to an acceptor molecule without emission of photons. That means you don't see emission or fluorescence of the donor molecule, but you see emission of only acceptor molecule, provided there is a FRET between the two, provided they are together. So how this rate of energy transfer? There are 3 important conditions to have this FRET happen. One is mutual orientation of the donor and acceptor molecule. This should be close to each other. That is why I said co-localization can be studied better with the FRET. Then another important thing is spectral properties of donor. Whatever is the emission of donor should be excitation of acceptor. Only then it will get excited and emit fluorescence. And third thing is distance between them. The distance between them should be less than 10 nanometer. This is how the FRET works. Like if you excite your donor and the acceptor is not close by. Now, let's first see what is the pattern of excitation and emission. Now, donor is getting excited by blue. The emission of the donor is in green. However, excitation of acceptor is in green and emission is in red. So now if donor and acceptor are far apart, the donor whatever donor is emitting as a green light will not be taken up by acceptor. But if they are close together, then there will be a transfer of energy. So we won't see green light, but that green light will be taken up by the donor, acceptor, and emit red light. So there are 2 important conditions: spectral overlap, as I explained here, and distance between this should be less than 10 nanometer. Now, if this FRET happens within the molecule, it's known as an intramolecular FRET. Now, this is an example. I hope you are familiar with CFP and YFP. So suppose it's a big molecule. At one end it's CFP and one end it's YFP. Now CFP is a donor, YFP is acceptor. Now if there is a change in the conformation of this molecule, CFP and YFP will come together and the FRET will occur. This is known as an intramolecular FRET. Suppose there are 2 different proteins, again labeled with CFP and YFP. If they come together, then there will be intermolecular FRET. So FRET is much better assessment of co-localization than the simple fluorescence color. Another technique that I would like to just talk about is a fluorescence correlation spectroscopy, or FCS. What this technique does is it measures the molecular movement in a small volume. It could be a cell, it could be a solution. So it helps to understand interaction of the molecules with each other. Now this is how it looks like. If the laser is passing through, this is the area where the movement is recorded. And then you can measure this movement and get a graph with time versus fluorescence. I'll just try to show a video for this. Now this is the area where it's going to record. So if there are different molecules, different size, the intensity will be picked up in that area. If two molecules come together or at a time, then we know that there is interaction happening, because there will be change in the signals that we get in this region. So most of the times this FCS is applied to look at the molecular interaction. It could be ligand receptor, antigen antibody, drug target, or change in protein conformation, Brownian motion, enzyme kinetics, diffusion, etc. Another important technique that I would like to talk about is a FRAP. It's fluorescence recovery after photobleaching. In this case, cells which are fluorescently tagged are exposed to a laser. The laser is parked at one position. What I mean by parking is not scanning to take images, but it is fixed for few seconds at one position. So this area is totally quenched, and what you get is something like this. Now, if there is a movement of molecules in this region, we should be able to see change in this black color. So then what is done after parking the beam and photo bleaching the area, you take images at different time interval. Now you can very clearly see that fluorescence is picking up, and after some time it's same as it was in the beginning. It is also shown as a graph. So FRAP actually examines mobility of different components within the cell. This is a movie of that. You see in this region, it is going to, yeah, it's blackened out. And now after some time, again, you will see fluorescence coming up, because molecules are moving from these two regions in this area of interest that we have been observing. So fluorescence recovery after photobleaching is important to study movements of molecule within the cell. So it is generally used to see diffusional mobility, to identify immobile fraction, to see the binding time that is required, the percent recovery that is happening. Now, another extension of this FRAP is FLIP, that is fluorescence loss in photobleaching. So what is done in this is, your area of interest is, say, for example, this membrane, and you want to see what is happening, what is the dynamics of membrane in this cell. So what is done is, there is another area that you mark as a bleaching area or bleaching ROI, and park the beam here. So you go on parking the beam, and so this area will keep on getting bleached totally. And if there is any movement from membrane towards this area of molecules, naturally there will be a reduction in fluorescence first in the neighboring area, and later on even the membrane will lose its fluorescence. As you can see here, it's enlarged in this. So over the time, even the membrane is so faint that you cannot see, but it tells you very important information that even in this area, some molecules from membranes are moving. So there is a good dynamics that can be seen by using FLIP. And lastly, I will like to talk about fluorescence lifetime imaging. Now, what is fluorescence lifetime? When we excite, say, fluorescence with a blue laser, it is sitting in the ground state. It gets excited and start emitting the light. Then it comes down to the ground state. So the time that it takes from excited state to come back to the ground state is known as a lifetime of the fluorescent substance. So instead of measuring fluorescence intensity, what is measured in this case is the lifetime of the fluorescence. That's why it is called fluorescence lifetime imaging. I had shown you, okay, this is a different picture. This is the picture where, and this is affected by the environment of the object that you are dealing with. So for example, this is lifetime imaging, both the pictures. One is in 175 millimolar, 174 millimolar sodium chloride, and another 500 is in 2 millimolar sodium chloride. Sorry, something went wrong.
Ma'am, I think one of the
I think,
participants has
okay, fine.
just, yeah.
I don't know. I got a pain somehow.
It's
I'll
fine,
just
ma'am. I think it has
I'll just
gone
take,
back to.
try to take a laser otherwise.
Sure. Yeah.
Okay. So in this, what you see is comparatively less fluorescence. Look at the intensity of fluorescence. So with higher concentration of sodium chloride, there is a quenching of fluorescence. But if you reduce the concentration, you get much stronger signal. So this is important to understand the environmental parameters like pH, ion, or oxygen concentration, molecular binding that is happening in the proximity of the fluorescent area. So this gives you information not only about the sample, but also about the environmental parameters that are present in that. This is another example of fluorescence lifetime imaging. I had shown you this convallaria picture. This is the picture where intensity of the fluorescence was measured. While in this, what is measured is a fluorescence lifetime imaging. So you can see much better resolution in this. More details are seen in this picture. It's the same field that is shown. I hope you agree that whatever you see in this area was not very clearly seen, these red particles that are present, but it was possible to see only because the imaging was done in a lifetime imaging mode. Thus, I would say that fluorescence microscopy or laser confocal microscopy in the fluorescence mode had enabled us to understand 3D world of nature, and it is an essential tool in basic sciences, and it's really an important asset in healthcare industry also. I hope now you agree with my initial statement that laser confocal microscopy has brought rainbow revolution in the biology. I really thank you all for your attention and patience. If there are any questions, I'll be happy to take.
Yeah, indeed, ma'am. I think not only the session, but even the slides and all were so colorful. I think with your clear and crisp explanation and with all the practical examples that you covered, I think it just made it more easy
Thank you.
to understand the session. We have now opened the floor for any queries. I don't see any queries in the chat box, but if any of the participants, if you all have any queries that you want to ask, probably we can spend a few minutes in doing that. If anybody has a question, you all can unmute and maybe madam can answer your query. So anyone who has a pressing query can go ahead and unmute and ask the query.
I think it's a tea time. They all need a good tea.
No, I think, ma'am, you need a break because I think constantly the energy with which you were speaking last 1 hour, I think it's just amazing, and the passion for the subject was clearly visible in your delivery of the session. So yeah, I don't see any query yet. Yeah. What I've also done is I have also added a feedback, link in the chat box. Kindly give us your valuable feedback so that it will help us in improvising all of our, future sessions. And what we'll do is I'll take this opportunity to thank Dr. Nishigandha Naik, madam. Ma'am, thank you so much for, first of all, accepting my our request of presenting over here, and for running us through this session and for sharing your insights with all of us. I'm sure all the participants have greatly benefited from it, and they'll be able to apply it in all their respective, research areas. I do see some reactions on the screen, so I'll take that as a positive feedback. So with that, just like to remind you all, I've shared the feedback link. Please do share your feedback with us. This session has been recorded and we are going to upload it on our YouTube channel, and I will share the link with all of y'all. So ma'am, if you have anything to say, maybe we can do that, or if
If
not, okay.
anybody has any queries and want to communicate, you can share my email ID. I'll be happy
Okay,
to in touch.
Hello,
No
ma'am.
issue.
Am I
sure.
audible?
Uh, yes, you're audible. Sorry.
Yeah.
I didn't get your name.
Yeah, my name is Tubai. So, in case of
Okay.
FRAP, ma'am, can you use that two color of two photo channel and check the diffusion of photo bleaching after photo bleaching?
I didn't get your question clearly. Could you repeat, please?
So you show now that FRAP is an experiment that fluorescence after photo bleaching.
Hmm.
So if I tag my protein with different parts with two different colors and focus the
Hmm.
laser beam in the two different position,
Hmm.
so can I check for different parts like the diffusion is happening where it is faster, where it is
Yes, yes, it's possible because basically what you're going to do imaging, you have more than one channel. That's it. That's possible. That's not an issue. More than two also, you can go ahead with that.
yes, yes, yes. Like we just have to tune the laser beam on according to that diode according to that
No, no, no,
fluorophore?
no. Laser beam is basically it's used for two things. One is to photo bleach. That is going to be constant for all of them. And when you're acquiring images, you have to use two channels instead of one channel, which will pick up your two different fluorescence that you're planning to use. That's it. Most of the time, just one single laser. If it's argon laser, then you choose
Okay,
your fluorescent substances such that they both can be
okay,
sort of
okay,
get excited. Even if you want two lasers, that's fine, but that turns out to be a little
okay,
expensive, but it's possible in principle.
okay, I got it. Thank you, ma'am.
Okay.
Yeah. Ma'am, there is one query in the chat box from Saili Joshi, and she
Mhm.
says that does excitation of fluorescent substance multiple time leads to photo bleaching? I'll
Sorry.
repeat that. She says, does the excitation of a fluorescent substance multiple times leads to
Hmm.
photo bleaching?
Yes, that's true. That's why when we expose the whole sample during laser confocal microscopy, even the planes which are not in focus could quench. They would bleach. So the more the exposure to any kind of light, they are going to bleach.
Yeah. I think this madam had covered this during the session, so I hope, Saili, this query has been resolved. Yeah, she says, thank you, ma'am. Yeah. Yeah. Any other questions that you all have, participants? I think this is a great platform. You're getting to connect directly with an expert with who has so many years of experience with this technique and much more. So I think please go ahead and make use of this opportunity. Think there are no further questions, ma'am. But as you suggested, I will definitely share your email ID with them, and probably over email if they have anything, we can stay in touch. You all can also reach out to us, and we can connect you all to ma'am and see how we can help you all with all your confocal queries. So with that, we'll wrap up this session. Thank you, madam, once again. And thank you to all the participants for joining on this Friday evening. And please stay tuned with our next session will come up next month. Details will be announced on our website soon. Yeah. Thank you once again.
Thank you all.
Thank you, ma'am.
Thank you so much, ma'am.
No, it's a pleasure. I'll sign out.
Yes, ma'am. Thank you, ma'am.